We're good? All right. Oh, I'm just waiting. Okay, cool. All right. I was asked to step in for Manu, who had, he spends a lot of time on the ocean. So he'll be coming and joining us Sunday night. Remember, we'll have a lecture from him at 7 o'clock on Sunday, and then the public lecture. Sitting in on the lectures of the past week, I've been trying to figure out a good transition from things that you've learned from this past 2 weeks to the next couple of weeks. So it's basically like a sort of a summary of areas of biomechanics inside cells that will help you understand some of the tissue scale processes, I hope, next week. And all the work I'm going to talk about today really sort of reflects stuff that we've been working on since I started my group. So this is a sort of hopefully almost encompassing set of people that I've learned from over the past 17 years. Okay, so what I've been really interested in, many people in the school, is understanding how the matter within cells at the cellular scale or at the tissue scale allows morphogenesis and force generation and shape change, right? So this is a movie of a single fibroblast cell, a mesenchymal cell that moves around and crawls and remodels tissue. And what we're looking at is a fluorescently tagged protein within the cell. This is the protein of myosin 2, a molecular motor protein. And these structures you're seeing here are actually assemblies of about hundreds of myosin motors within the cell. And you can see as the cell is migrating up the screen that these structures are assembling at the front of the cell, disassembling at the back of the struct. There's strains within the cell, and the structures are all moving around, right? So there's both force generation that's occurring within these networks, but then also a lot of remodeling that's occurring. And so, starting about 25 years ago, 30 years ago, cell biologists started identifying all the different proteins that are in the different kinds of physical machinery within adherent cells, okay? So this is another kind of immunofluorescence way of imaging the polymer networks within the cell of the actin cytoskeleton in blue. And this is my physicist rendition of all the structures that are these contractile networks, these actomyosin networks. We're going to be talking about those a bit more, that are generating contractile tension. At the front of the cell are polymer networks, also built out of this biopolymer actin that is generating protrusive forces that are pushing forces at the cell front. And then there's organelles that couple adhesion between the cell and the extracellular environment that again assemble and disassemble. Okay, so even 20 or something years ago, all the molecular machinery that governs these sort of physical structures were well identified. Okay, but what I still find fascinating, is that as all these stresses are building, these physical structures are assembling and disassembling, the cell still just smoothly walks along. It never rips itself apart. It never undergoes some catastrophic deformation, right? It's able to do this very smoothly at all the different scales, right? This is one of these key aspects of biology, right? At all the different scales, biology is able to change its function, its shape, its motion, and really doesn't do anything like, like gelation in a strong, and the way we think about it from a physics view, right? It's able to smoothly change its shape over all these time scales. Okay. So the way sort of we think about all this squishy stuff within cells is that there needs to be these nested systems of physical and chemical machinery that are constantly sensing physical and chemical forces and stresses. There needs to then these feedback to modify its own mechanical response. And this also controls how forces are generated. Right. So usually, like for much of my life, I like to stay in the mechanical response regime or force transmission regime, like force generation, but really to think about how biological systems and living systems are evolving, you have to think about this feedback. And we've been starting to think about these feedbacks thus far. If we go back even up beyond the sort of the physical matter or the living matter stage, I spend much of my time thinking about how cells function physiologically, like what their biology is, right? And their cells also have to sense cues from their environment. They sense chemical cues that tell them to grow, right? So chemical stimuli that say like, okay, start transcription or start translation, right? So they sense all these physical chemical cues from their external environment, right? And this drives their migration, their movement, their motility, like Kirsty was talking about, right? Tells them what kind of cells to become, bone or muscle or fat, right? So all these at all these different scales, this matter within cells has to be sort of sensing and processing all this information. Okay, so depending on who you're talking to at any given moment, right, you can think about these questions with different lenses on, right? You can think about it from your physics perspective as a sort of active and adaptive soft matter, right? You can talk to cell biologists or developmental biologists and think about how do you process information at the cell or tissue scale to control your physiology, right? If you're thinking about information theory, you can think about how information is processed in this, but in contrast to something where there's like software and hardware implementing the cues, right? In this case, the information is processed by the physical material that you're sitting in, right? And so this is an emerging field that you'll be learning a little bit about going on of like physical learning of like how these disordered materials are actually processing cues to change their form and function, okay? And all of this requires us to think about mechanochemical feedbacks, right? So conversion of information between chemical and mechanical information. Okay, all right, so we're gonna start at the most simplest scale. So the way I'm organizing this talk, and I should just, wonder if I should go to the organization here. I'll just start here. Is I'm going to start from the basic components of the polymers. And so we're going to think about this within a lens of how do you process information and store information and transmit stresses within these polymer networks. And we're going to start at the simplest scale. I'm not going to get very far in this 90 minutes. Hopefully we'll get to the point where we have some activity and we'll stop when we don't have any more time, okay? So Mo, I think, introduced a little bit about semi-flexible polymers. You heard a little bit about semi-flexible polymers from Kirsty. So just as a reminder, these polymers that are in cells, actin filaments, intermediate filaments, microtubules, are all semi-flexible. You can reconstitute those outside the cell by grinding up tissues that have a lot of actin polymer and grow them in biochemical reconstitution. So here is a fluorescence image of an actin polymer that's just been bound down to a surface of a glass cover slip. And you can see the contours and estimate the persistence length just from looking at the tangent-tangent correlation vectors along this polymer. And the ratio of the bending modulus to thermal energy gives you a persistence length, right? So for actin filaments, it's on the scale of 17 micrometers. Microtubules are much more rigid. Their persistence length on the order of a millimeter. Intermediate filaments are much softer and have a much shorter persistence length, right? So just in comparison and just to give you some sense, you know, semi-flexible polymers are not very ubiquitous in our sort of other kinds of soft matter. It's actually quite hard to endow semi-flexibility to other kinds of synthetic polymers. So if we think of polyethylene, right, this is a very canonical flexible polymer. There's no bending rigidity between subunits. And so here, the decorrelation length happens on the scale of individual subunits. And so the effective persistence length is on the scale of 1 nanometer. Okay. And so this idea of how you characterize a spring constant of semi-flexible polymers was of interest, I don't know, 30 years ago or so. This is a schematic summary done by a former student, Patrick McCall, where you can actually take optical tweezers, pull on two ends of the polymer to measure a force extension curve. Now here's where I'd want a board. So the way we think about polymer elasticity is think about the entropic degrees of freedom available to that polymer. And so if this was if we imagine two tethered points on this polymer, we imagine the amount of thermal fluctuations available to this polymer, right? And we can think about it in the context of sort of like a reptation tube. So this is like, you know, one of these slithering snakes of thinking about the fluctuating degrees of freedom. So when there is little force on this polymer, that the amount of fluctuations that thermal forces the amplitude of those transverse fluctuations is large. The entropic degrees of freedom is bigger. As you pull extensional force, the amplitude of those transverse fluctuations decreases, and that gives you a higher entropic elasticity. So as we apply an end-to-end extensional force on this entropic spring, here we're pulling out here at this early stage of these really low forces, an entropic elasticity. And then as you pull higher and higher, right, you get to this enthalpic elasticity. Now, just look at the increased spring constant of the enthalpic elasticity of this polymer. Like, if you think about protein polymers in the absence of thermal forces, they'd be like a hard plastic, right? So this is the enthalpic elasticity. But with something like a semi-flexible, it's very soft initially because you're pulling out those entropic fluctuations, and then you start to diverge and approach the enthalpic elasticity limit, okay? A few lectures ago, we thought about Euler buckling. Because these are flexible, now if you compress it, you get Euler buckling very easily. So, stepping back, you know, you can think of these as having under extension, having two different kinds of spring constants, a sort of soft entropic spring constants that diverges and becomes a nice stiff enthalpic spring constant. But under compression, it's very, very soft, okay? I also want to encourage any questions that come up, okay? All right. Okay, so that's just the semi-flexible polymer. The semi-flexible polymers are so cool. All right, I just want to get you excited about semi-flexible polymers. Yes.
Bending modulus that you get from looking at thermal fluctuation and the one that you get from tweezing the microtubules actually come out very different, and like people don't understand why that's the case. I'm wondering if actin is a similar story.
Actin, that's interesting. I didn't realize that about microtubules. Huh. Well, how different are they? Like, so 1 milli, like which one's stiffer? Interesting. I mean, there is a concern. I am not aware of that with actin filaments. I mean, microtube, that is surprising to me. I would love to talk about that further. I didn't realize that. No, with actin, I think the biggest difference in terms of the bending stiffness, first of all, when you're doing these measurements experimentally, there's always an issue of what you measure as a curvature when you like stick the filaments down on a surface. That sometimes gives you a good approximation of thermal forces, but it's not as good as just measuring the actual fluctuations. But also, like, what other proteins are decorating the outside of the actin filament makes a big difference. Okay. So the other thing I mentioned here is that also in contrast to synthetic polymers, these all biological polymers are enzymatically active. Not so, all is probably an overstatement, but actin is. Actin and microtubules are. So they hydrolyze chemical energy to assemble. Okay, so they're a mechanoenzyme in their own right. Okay, and what that means is that when you polymerize the actin polymer, it is asymmetric. So this dark red portion of the polymer is what is sort of one end. It's called the barbed end of the polymer, where your subunits that are assembling are chemically distinct from those at the other end. And there's a hydrolysis of those components along inside the polymer. So the polymer itself is hydrolyzing these subunits and setting up a chemical gradient along the polymer, okay? So since you have a chemical gradient, those two ends of your polymers are different from each other. And that means that at one end, it can do work, okay? So when you polymerize these polymers, because of their semi-flexibility and the fact that they are mechanoenzymes, they generate forces through polymerization, okay? And also that chemical gradient along the polymer sets up a way to regulate other kinds of binding interactions along the polymer that can also set up asymmetries in the larger scale networks that you assemble with each other, okay? So for instance, there's gradients, there's other polymers that are also regulating how these polymers disassemble. So that's another way of relaxing mechanical stresses and how the polymers are assembling. So once you start adding more and more components, you can set up larger types of chemical asymmetries in these systems that allow you to generate different kinds of work and build up stress and relax stress in different ways. Okay. All right. So with that, that sort of control over polymerization, when we think about a kinetically arrested actin gel, I think, again, Mo introduced some of these ideas to you in her lectures. What we imagine doing is we take a collection of actin monomers, the subunits, and then some fraction of cross-linking proteins that can cross-link filaments to each other. And we assemble those together. As this whole system is assembling, the polymers are growing in length, and then as they grow in length, they start to have steric constraints with each other. So they start out as small objects that can rotationally diffuse in the solution. It's just a solution of short filaments. As those filaments get longer and longer, they start to overlap with each other, and then the cross-linkers, they can become sterically entangled with each other because they can't physically cross each other, but also the cross-linking proteins stick those polymers into place, okay? So then you grow into a gel, a network that is a kinetically arrested object, material, okay? Before we just go into the kinetics of that, all these cross-linking proteins that you find inside cells, these are schematics of them. There are some that are very short, a few nanometers, and very rigid, but then you can extend the length of these. So this is one of my favorite cross-linkers. These are the blue domains are where it binds the actin polymers, and the intermediate sections it's just basically a big flexible polymer. So it's like, actually, it's rigid regions and also flexible regions like my elbows and my shoulders. So it's like I'm cross-linking two actin filaments but can wave around. So it's very flexible and allows for a lot of rotational flexibility. Yep. Can you repeat what triggers the polymerization? In this case, salt. Yeah. So salt is very good. You can also add other proteins that can trigger the polymerization. Okay. Okay, but the important thing is like, whatever structure you get here, as we show, well, I'm going to go into the next slide, I think. Oh, I was going to go through my outline in the next slide. Is going to be dependent on how fast you polymerize, like how fast those rods are growing versus their orientational diffusion and the nature of all these cross-linkers here. Okay. Okay, so see, this is my organization. Sorry about this. All right, so I'm going to talk about first what kinds of matter you can get simply from that two components. What happens when you get polymers and cross-linkers, and just go through the kinds of soft matter you can get there. And then I'm going to ask, what are the consequences of adding more mechanoenzyme activity? And I'm going to focus on molecular motors that drive sliding between the polymers, not so much on the assembly and disassembly, okay? And I don't think I'm going to get to number 3, but it's sort of my passion at the moment. So I love to talk to people about this, is like, how do you trigger feedback of force-activated response? What object sense tension to then modify the response, okay? And I guess I want you to think in your head, I think, you know, I'm going to talk about a lot of things. The things that I think are the most interesting in the field currently are really about when we set up these systems, especially these simple soft matter systems, how do all these configurations store mechanical information in how they're built? How do you generate patterns and structures? So we've talked about flocking, we've talked about fluids, right? How do you get patterning in these kinds of active driven systems? And at what points can you get new material properties? And the reason I chose to start with these simple systems is because you don't need that much to get really interesting phenomena occurring, as you can already see with some of these active matter systems. And I think it's really interesting to explore just the really sort of basic systems of where you get that kind of structure. Okay. All right. Okay. All right. So for the next for this slide, just ignore the crosslinkers. So we're just going to go back to pneumatics, like what Christina discussed before. So if you just have your rods, your actin filaments, your rod-like polymers, and imagine a space of concentration of those rods. So the rods are a constant length, and you're just changing the density of those rods. As you increase the density, and when the distance between those rods is very large compared to the rod length, right? They are just a very dilute solution of polymers. It's just a simple fluid with rods. Nothing interesting comes out. As soon as the distance between those rods becomes smaller than the length, those rods start to become entangled. This now becomes a semi-dilute solution of entangled polymers. Then you start to have viscoelastic response. You start to generate elasticity in those solutions of polymers because you're reducing the entropic degrees of freedom of those polymers themselves. Okay, so that's semi-dilute solutions. And that's still, for something like actin, it's still, this is in micromolar. I think this is a concentration. But the length scale here, so if lengths are on the order of like 2 micrometers up to 20 micrometers, right? The mesh sizes, because again, the semi-flexibility of the rods is on the order of about, half a micrometer up to 3 micrometers is a typical mesh size. Inside cells, that mesh size can get smaller, but we're thinking about things that you can resolve with high resolution confocal imaging. Okay, so these are semi-dilute solutions. As you increase the concentration of your rods further, then if you go up 3 orders of magnitude, that's when you start to get dense solutions, and you can go through isotropic pneumatic phase transitions. All right, so this is something that was, I think, first characterized by Paul Jamney and Jay Tang, about 3 decades ago. So they started to look at under cross polars to see pneumatic transitions of these actin nematogens, these actin filament nematogens. And so what you see is if you drive a dense solution of actin, you start to see the appearance of these pneumatic phases, which appear as sort of these things called tactoid-like droplets, which is this ordered phase within this disordered liquid phase. Okay, but this is at concentrations of 1,000 micromolar, so very much higher than most of what we're studying in sort of traditional rheology of entangled networks. This is an image from my group of a pneumatic phase of an actin filaments when you crowd it down in 2D, which is the way we were able to get to that high concentration of actin. Okay, so you can start to see those +1/2 defects that Christina had pointed out in the microtubule-based system. Okay, so just by changing the concentration of your rods, you can drive your system through these different states. Yeah. 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 So what happens if you have so this is for short filaments. Yeah. What happens if you have longer, you basically, it's hard to get to pneumatic phases because everything is kinetically arrested because the relaxation times get too long to be able to actually get a nice clean pneumatic phase. It's not quite a polymer melt, I don't think, but it's definitely the relaxation times get way too long to get a really clean pneumatic phase. Thanks for asking a question. Yes. Yeah. That's a great question. We'll talk about that in a little bit. Yeah, so that shape is determined by the surface tension, the sort of the isotropic surface tension, which is driven by the liquid crystal elasticity in that case. And we'll definitely talk about that in probably a half hour or something like that. Okay, good. Yeah. Okay, sorry, can you? No crosslinker. These are just rods, like just filaments, polymers. So what does the entangled solution mean? Yeah, so it just means that there's polymers in 3D, let's just say in 3D, where the filaments the length of the filament is greater than the mesh size, the average distance between the polymers. And so there's steric constraints. And as soon as there's steric constraints, that sets up a reptation tube, Like it basically sets up reduces the entropic degrees of rotational freedom of your polymer, and it gives you a viscoelasticity. And I'm happy to talk about that at length. Yeah, so it gives you, when your length is shorter than your mesh size, then you don't have any elasticity in your system. Yeah. No, not yet. So this is all high density. So look at this, like this is at one density. This is a very small concentration difference. And this is at the high density of actin filaments. And then there's a critical, the transition to a liquid crystalline phase where you start to prefer ordering. So this is not in a gas. This is a very dense, disordered liquid to a dense liquid, ordered liquid phase. Yeah.
Get these tactoids and maybe increase the concentration more, and then it's somatic everywhere.
Presumably, yep.
Like, does something interesting happen to the size of the tactoids as you go through that transition, or is it like you get a bunch of tactoids and eventually they fill all the space?
That's a great question. Do you know, Leo? I actually have never studied tactoids. The question is like, what does it look like when you go from a nucleation of the liquid crystalline phase? Like, how does that grow? I actually don't. I'd have to look in the literature. Like what does the dynamics look like from the nucleation of the pneumatic phase to a continuous pneumatic phase? It would just be it would grow. Yeah. And there'd probably be very limited. It's not going to be diffusion, or it's going to just grow. Yeah.
Is that stable at that concentration, or is that just what?
Is it like a kinetic transition? That's a good question. That could be an open question for next week. Yeah. 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 That's another good question. I think there's a big literature on this. I am not aware. Yeah, I think if I went back and look at Jay's and and Paul Jamney's papers, I could help you look for that. Yeah. Nothing, just actin filaments. Yeah, just Oh, no, sorry. Actin's always in a solvent. There's always an aqueous background. And so then, so you can get these high densities in multiple ways. Actually, you can add crowding agents, so that go into the liquid phase, and so to make a polymer dense phase. So that's sort of what we do here with the 2D. We add crowding agents that push everything down to the surface. If I recall, I don't think Jay and Paul did that. I think they really just concentrated their actin filaments that high, I think. But they might have also used a crowding phase. The concentration, yeah. So this is, imagine this being done with a constant length polymer. Yeah. And then as you change your length, this transition would happen at different concentrations. Yeah. So just imagine this concentration happening at one length. And then somebody added the other caveat that this pneumatic phase wouldn't really happen if you made your polymer lengths too long, right? Because everything gets just too long. Okay. Thank you for asking questions. All right. Good. Okay. So we're going to leave this. This is just sort of brief sort of space of just actin filaments. So when you watch gelation occur, gelation is a kinetically arrested structure. So whatever gels you form are history dependent on the initial conditions and the assembly conditions that you form. Okay, and that's true with any gels. But what's super cool about, again, actin networks and other biomolecular networks is that you can control those kinetic rates through other kinds of biochemical factors. Okay, so this is a movie that a previous student of mine took of an actin gel just forming. So over the course of an hour, you can see sort of rotational diffusion of small bits of bundles. Those bundles grow in length. And at a certain point in time, those bundles stop moving, right? Everything stops moving because everything that's the kinetic arrest, okay? So you have diffusion, everything's growing. And so schematically, you can go through and do this by simulations or whatnot. You have these short polymers. The polymers are growing in length, and then at the point when the length becomes bigger than the mesh size, that's when the kinetic arrest occurs. Okay. What? The polymer length can be much bigger than the mesh size. Yeah, and the mesh size is determined by the 1 over the square root of the monomer concentration, not the sort of the length of the polymer. And it's because you can control the length of the polymer independently of the mesh of the concentration by adding other kinds of proteins. Yeah. Yeah. When the length becomes bigger than the mesh size. Yeah. Okay. Okay. So I'm not going to go into any of the molecular detail of this, but each of these things, right, of how likely it is that you're going to get stuck when you get bigger than your mesh size, that's going to be determined by your cross-linking concentration. How fast you're growing is going to affect the time scale at which you're going to become kinetically arrested and your overall density. And there are proteins within the cell that control all these factors, okay? So you can control this kinetically determined state by the same concentration of actin polymer, because you have control over the biophysical determinants of the length and how they're arranged into bundles or mesh size. You can control this structure. And this structure is all going to determine the mechanics of the resultant system, right? This is not, there's nothing in equilibrium here, right? This is all, and so it's important, everything how you prepare the system, how the experimentalists prepare the system, and is going to determine what your resultant mechanical response is. Okay? Okay. All right, so once you have a gel with the cross-linkers and the networks, then if we want to ask a question like what the mechanical response is of the system, we usually use a technique called rheology. Has rheology been introduced to you before? Did Mo talk about it? Yes? No. Okay. I see one no and one yes. So, okay. All right. And then everybody else is blank. Okay. So what you do is, like, again, an experimentalist, you go into the lab, you have these two solutions, you prepare a little gel, which is around a small volume, about 200 microliters and then you put it in a device that has a torque control. So you can measure, you apply a torque to the system, and then it reads out how much displacement has occurred. So that is a shear stress, a force in a 1D, and you're measuring how much distortion occurs. You know the height, the distance between those two plates. So the ratio between the displacement, the horizontal displacement, and the height is the strain of the system. That's how much distortion has occurred, the shear strain. And the force that has applied, that you divide that by the area of that top plate, the force over that area is the stress. And the stress divided by the strain then gives you the elastic modulus, okay? And so now I know Noah talked about this before, so this is now a reminder that when you have a solid, right, the amount of stress you apply for a pure elastic solid is proportional to the strain. That is a measure in shear stress of the shear elastic modulus. In a fluid, now a fluid is measured by a viscosity, which is just now dependent, the ratio of the shear stress over the shear rate, the strain rate, sorry. And so the how fast you're just distorting that top plate, okay? Now, most soft matter is viscoelastic, so that means that it has characteristics of both elastic solids and viscous fluids. And so what we do there is measure a frequency-dependent viscoelastic response. So by applying an oscillatory shear stress or shear strain in this case, and measure a frequency-dependent elastic and loss modulus. Okay, so we measure things at different time scales. Okay, so one way of visualizing this, if you look in time space and not frequency space, would just be doing a amplitude of a constant shear stress. So in this case, the red line is a constant shear stress, and you can measure out what the strain response would be of that system. So the black line here is what, so for a cross-linked network, the strain would evolve over time. Now, then you take off the applied stress, and the strain recovers partially, okay, over some time. You can also visualize this now in frequency space. So instead of time, you can do a Fourier transform of this and measure a frequency-dependent elastic and loss modulus, okay? So here, if we look at high frequencies, these are short time scales, And also the short time scales are also measuring the short length scale frequency dependent response. Typically, again, for these polymer networks, when you're looking at any frequencies above about 5 hertz, you're really measuring the bending modes of the polymer within that crosslink network. At time scales below about 5 hertz, what you're starting to measure is, again, if you have a polymer, how much that tube, that reptation tube that your polymer is in the network can change. And that's going to be dominated both by sort of the steric constraints that you're in, but also the physical constraints of that crosslinkers that you're within. So that's telling you how much that polymer or that bundle of polymers is able to relax. And that really long time scale is probably even lower than 0.01. That's really measuring your reputational diffusion of the whole bundle along its tube. So that's giving you long time scale relaxation. So as you're looking in frequency space, you're looking in both short time scale and length scale response. And at longer and longer, lower and lower frequencies, you're looking at longer length scales, longer time scales. Okay. So you can see here that this is one measurement that was done by, I think, Dave Waits's group, where it was the same network, and all they did was measure the temperature dependence of a cross-linked polymer network. And what you see is that at the lowest temperatures, the elastic modulus, it dominates over this viscous modulus over the entire frequency range. Okay, that means it's predominantly solid over that full time scale. And at the highest temperatures, now the elastic and loss modulus are similar magnitude, and it's also changed its slope as a function of frequency, meaning it's more like a viscoelastic fluid at that point. Okay. Now, the way we think about this temperature change is, again, I told you between these regimes, really the mechanical response of these systems is being dominated by how fast the crosslinkers, that polymer that's constrained, is being relaxed by crosslinkers. So these biological crosslinkers, also something cool that is really sort of unique to biology, is that the crosslinkers have off rates and dynamics that are on the time scale of seconds and are they have time scales. They have off rates on the time scale of seconds, which means that you can relax stresses at sort of this nanometer scale pretty quickly. And so when you unbind this, what you're measuring is a temperature dependence of this off rate of the individual crosslinker and the collective properties of the relaxation of that whole crosslink polymer network. Okay? Yeah. Oh yeah, so yeah, I'm obsessed with shear moduli for some reason. Yeah, no. No, you're absolutely right. You could, so this is all shear elasticity. You could probe, if you did a mechanical measurement, other dimensions, you could absolutely measure the other types of elastic moduli and ask the same exact questions. Yeah, this is just a very myopic view of shear elasticity. Yeah.
The size of the cell that you use to
Absolutely, yep. Yeah, that's, as an experimentalist, you have to be really certain that it is size independent. That it's an important test to do when you're doing experimental measurements of rheology. That whatever measurement you're getting, it is independent of the gap thickness. Yeah, it's a good question. Yeah. Oh, it's all liquid. Sorry. Yeah. Everything is an aqueous solution with some osmolytes, like some ions, some ATP. Yeah, there is no air ever anywhere except for at your skin. Yes. Yeah. Okay.
Like, there's some kind of interest in it.
Here, let me see if I can. Okay, so here, okay, I said this 10 hertz right here. So, okay, what I want you to imagine is all the elasticity. This elasticity is all driven because you're reducing the entropic degrees of freedom of your semi-flexible polymers. Okay, that's what all this viscoelasticity is coming from. So your polymers would like to have lots of rotational degrees of freedom. They would like to have lots of thermal fluctuations along their length. And when you pin them down by crosslinkers, you're reducing the number of entropic degrees of freedom that you're giving them. And the length scale at which you're pinning them is setting a maximal length scale at which they can fluctuate. And then when you relax one of those crosslinkers, that's allowing another degree of entropic sort of bending mode to propagate. So at the highest time, so let's just say the crosslinkers had no off rate. Okay, they were just pinned at a certain length scale. What you would see is that this rheology would go up. It would look like it has a characteristic scaling with frequency. That's just the fluctuations. It's omega to 3/4 for anybody who cares. Fluctuations of the polymer modes, because you're just looking at a pinned piece of rope like this. That's fluctuating, and depending on the length scale at which it's pinned, it's going that crossover to where you're gonna dampen all those that bending fluctuations is gonna change. So now if you have a crosslinker that's coming on and off, you're letting bending fluctuations that are longer and longer length relax. And then you also have these weird sloshing modes where so when you have entangled semi-flexible polymer solutions, you can approximate it in old polymer physics degen language by a reptation tube. And so that it's basically that frequency at those intermediate scales is changing in conformations of that reptation tube. That can only happen when you have crosslinkers that are unbinding. And so the crosslink unbinding, I mean, there's lots of physics that's going on here. The crosslinker unbinding is telling you about how those polymers can rearrange. Yeah. Yep. Yeah. So again, there's a lot of this is like a decade. I'm happy to go into more depth about this. This crossover frequency is sort of giving you a feeling of when this polymer can have full sort of translational flexibility in its reptation tube. So it's getting to the fluid state of the semi-flexible polymers is giving a translational mobility in that tube. That's this red one. So that red one has that crossover to lateral mobility is happening there because the crosslinkers are coming on and off so quickly. Okay? The other ones, because the crosslinkers are more bound, it happens at a much lower frequency. I believe with this data, I know with other data it's like this, you can rescale all this data. You can shift these in frequency and magnitude and get characteristic relaxation times to understand how that reptation relaxation is occurring. Okay? Yeah. Oh, a normal, let's see, a normal liquid would have in this case, because there'd be no elastic modulus, right? And it would be a constant viscosity. So then, so if we go back here, this would be the fluid, and let's look in here. Here, if a normal liquid on the left-hand scale, right? If we apply a stress, the strain would increase linearly with time. It would just be a constant strain rate. Okay, and then for a pure elastic solid, the black line would just be another step function back. Okay, yes? Oops. Wait, what'd you say? Is 15? I don't know. Do I want to put liquids near to freezing? I don't know. I don't know. I don't know why it's 15, not 4. Yeah, I don't have a good answer to that. I don't think so. I think it probably has more to do with physiological stability of the proteins. Yeah, I don't think it has anything to do with physics, I'm guessing, but I don't know. Yes. I hated spring dashpot models for so long. I can't tell you, and I've grown to be okay with them. I've grown. Oh, it sucks. It totally sucks. It's totally bad. Yeah. No, because you actually have to calculate the dispersion of bending modes. And so you can get, if you add enough dashpots and springs, you can get something that gets the frequency dependence, but it is not capturing the physics at all. No, it's awful. It's awful. Yeah. Sorry. That was like, yeah, my physics snobbiness. But then I realized spring and dashpot models can be useful. Sorry. Anything else? Yeah. Oh, yeah. So, okay. So G prime and G double prime are linked because through Kramer's Kronig. This has been like 15 years since I've thought about this. So they are dependent on each other. So when you have a flatter G prime, right, the G double prime goes to zero. Like when it's more elastic, the G double prime is going to zero. So the blue, let's just take the blue for example, that is a flat, it's almost elastic at those higher frequency range, and G double prime is very low because the ratio of the elastic to loss modulus, take the ratio of the filled into the circles is very high. So it's predominantly elastic. And then when they become closer in magnitude, then that becomes more viscoelastic. I'm not sure that's answering your question. Why? So let's just take this blue one. What's happening is it's transitioning. The frequency dependence here is predominantly elastic, meaning the elastic modulus is dominating over the viscous modulus. And as you go down in frequency, now it's becoming predominantly a viscoelastic fluid, and so they're becoming more similar in magnitude to each other. It's become a fluid, yeah. Yeah, because there's relaxation. Now, I'm not showing you the highest frequency. There's a whole literature about what's happening at much higher frequencies. This is still pretty low frequency. And there, you're really just looking at the bending fluctuations of the actin polymers. And then it actually scales like frequency to the 3/4, both of them. And so there's this wonderful work from, I think, Christoph Schmidt about 30 years ago that measured all that. So there's like, yeah, you can distinguish between semi-flexible polymers and flexible polymers by their high frequency bending modes that way too. Okay. All right. Okay. So I told you earlier that semi-flexible polymers are so cool. They're so awesome. So it's because, now remember that spring constant that goes from like the entropic elasticity. So that spring constant, if I go back, let me just go back to it really quickly. It's highly nonlinear. So even without going between the entropic and the enthalpic limit, let me go, where is it here? The spring constant basically starts to diverge earlier. So this is the enthalpic limit of the spring constant. This is the soft bending, the entropic spring, the linear portion of the entropic spring. But this is when it starts to go through nonlinear stiffening of the spring constant is nonlinear with respect to extension. Okay, so that means even in this entropic spring network, as you pull on these polymers, if you're pulling out entropic elasticity, the individual springs are nonlinear. Okay, and so you can see this instead of doing a frequency dependent measurement of doing an amplitude sweep. So that's what this is. So this is now a rheology measurement where you're changing the strain, the distortion, and measuring the stress response. And so here you see in the middle, there's a linear response where where the stress is linear to strain, but at the end, the stress diverges at the higher strain. Okay, so just follow the first track. So up here, it's linear, then it goes up. That's a nonlinear stiffening because the modulus, the ratio of the stress to the strain, is increasing. Here, I'm going to show you the next slide. So this is now a plot of the local slope. K is the local slope of the stress strain measurement. And so this is the linear regime where the slope is constant and then it diverges. Okay. So this differential stiffness of these networks is strain dependent. Okay. And if I just imagine, again, following the top and the bottom, it diverges if you go to the right. And also, here, let me just get this, right and to the left, okay? And basically the way you can think about this nonlinear stiffening is that as you shear the whole polymer network, it's this whole disordered network of polymers, you're pulling on your modulus, the stiffness of that whole network is dominated by stretching of the polymers, and you're probing the nonlinear stiffening of those entropic springs in the system. Okay, now you probably have noticed this, this gap between, so when you do one of these curves, you're going up in strain and then coming back down in strain and going back through. The area in this portion of the curve is giving you your viscous dissipation in your system. Okay, so that's giving your loss of the system, how much viscous stresses you're losing. Okay, but I want to right now focus only on the elastic, the strain dependence of the elastic stress, where it's nonlinear stiffening. Okay, and the theory here, this solid line is theory. This is the linear regime of this entropic spring is really the theory from the nonlinear extension of the entropic elasticity. Okay. And so that's been tested by many groups in many different situations to show that it accounts for the nonlinear stiffening. And again, like, so if we go back to my lofty goals of trying to think about like force responses, this is a system where now each strain, you're going to be probing a slight different differential modulus, and you're able to, this material is able to stiffen depending on how much extension it is. It's not, the inherent nonlinearity is pretty unusual for a type of material. Like jamming and sheared packings is another example, right, under compression, but like these polymer networks inherently give you nonlinearity. Okay. Okay. Now, because again, these, yeah. No, don't apologize. This is good. I'm just not going to get to motors. Okay, it's fine. It's okay. Which one? Yeah. Yeah. That you mean the dissipation part? Yeah, I mean that's a something I like to think about. I think that, I mean, there's so many things that are happening in cells to right, because cells are not in these kinetically arrested gels. Okay, so there is so much dissipation going on. I think basically at every single time point that, especially if we think of actin-myosin networks, there's stress being built in the system. It's constantly in this nonlinear regime, yet there's constant mechanisms of dissipation and turnover. So I think cells are always at this point where they're building up stress, relaxing stresses, and that the fact that they're able to sense when those stresses build up and turn over is like key. Like I think it's always in that nonlinear regime, right? But it's always also dissipating. So yeah, that's my current feeling. I don't know if it's true, but that's my feeling. Yeah. 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 I mean, this one is taken from like one of the branches. Like it's basically taken from like this curve. Yeah, we don't take into account the loss part of it. So what'd you say? Yeah, you can imagine doing it at low speed. We don't take into account the loss there. Yeah. Okay. Okay, so this is, I told you that in this case, the stiffening is dominated by stretching. Okay, now the other thing is that, again, because these are semi-rigid filaments, if you back off the concentration of how many rods you have, think of a very sparse collection of rods. When you shear that system, how your strain propagates to that material changes. So at really high density, think of, again, a crystal or a very dense polymer networks, the macroscopic shear that you apply it will be self-similar at all scales. So it's affine deformation. But when you start to have just few connections between these polymers, now the shear you apply at the bulk scale or the larger scale is not propagated uniformly, and it's highly non-affine, meaning that there's deformations within the network that are not self-similar to what's happening at the larger scale. Okay, so what happens when you back off your concentration? And if we're somewhere, what am I doing here? I'm over here. Sorry, I don't know why. If you're around in this area, so if you back off your length or you back off your concentration, you get to regions where you're basically very weakly connected. And I think Mo did talk about this quite a bit. Like this is connected to percolation, where you start to be dominated by bending modes in your system. Okay, and when you're dominated by bending modes, you can't get any of this stretch, and we can't get any of this nonlinear stiffening anymore. So what happens to your material, if we look at our now shear elastic modulus, this is the shear stiffening regime where the elastic modulus increases as a function now of applied stress. But if you get into your system close to the percolation limit, right below it, you start to get shear weakening. Okay, so just by small changes in your length, your bending stiffness, and your concentration, you can go from something that is shear weakening to shear stiffening. Okay, which is also a nice knob to control in terms of tuning how your force responsiveness is. Okay, yeah. You can change a bunch of different things. You can change, in this case, you can change your concentration of your rods, you can change your length of your rods, and you can change your bending stiffness of your rods. All of these things drive you closer to being dominated by bending. Yeah, yeah. Yeah, I broke it. Sorry, I was a grad student when I took that curve. Just rammed it. Okay, cool. All right. Okay. All right. So I've taken you through this this regime of now a state space. There's no like real phase transitions happening here of just actin, like a semi-flexible polymer of a certain length and a certain type of cross-link concentration. Okay. So we're in a place where we're in these kinetically arrested gels, and we're changing the cross-link concentration or filament length and going between things that shear stiffen or stress stiffen and stress weaken. Okay, so you already get a lot of knobs to modify. The other thing I didn't emphasize a lot is that when you're in this, the entropic elasticity, in addition to being highly nonlinear, the semi-flexible polymers, the spring constant of your polymer is very sensitive to the length of the polymer. So in the length here, what it means is the distance between your cross-linkers. So basically by pinning down the polymer at shorter and shorter lengths, you can get a much more stiffer, it's a highly nonlinear increase in stiffening of that semi-flexibility, which is also super cool. So it's a really tunable material just in those ways. Okay. Oh boy. Okay. Because I promised to talk to you about memory, I'm going to tell you about one kind of memory we can get even this really simple system, okay? And that is right sort of at this transition between stress weakening and stress stiffening. Okay, so I introduced this idea of stress stiffening, which is basically, you change your strain, you change your stress, and you look at the strain response. Now, in a stress stiffening situation, it's completely reversible. It's you put on your stress, you take it off, and you get the same network back and forth, right? It's a completely reversible process. That is in contrast to something known as work hardening, where you basically apply a stress or a strain to a system, and over time, the mechanical response to your material changes over time. This usually is caused because you're inducing structural rearrangements in your material that change your microscopic modes of deformation. This can happen in crystals where you're changing the amount of line defects that increase your work hardening. Andreas Bausch's group has also seen this for actin networks where you're applying stress and you can modify your mechanical response. This is basically as a function of number of cycles that the stress at a certain strain increases, and then plateaus off. So this is a completely irreversible process. a few years ago, a postdoc in the group wondered what kind of mechanical response we could get in sort of simple cross-link networks where we apply a stress for a certain amount of time. And so we do a readout of like one of these these differential modulus as function of strain curves, and then apply a stress for a pretty long time, like 400 seconds, which is much longer than we normally do a rheology experiment for. And then do a readout that's very similar to the cycle that we did before the stress application. And what you can see is that now there's an asymmetry in the nonlinear response in the system and this is after this applied stress has been removed, okay? So that would look like work hardening, right? That would look like something where you apply stress, then you take off that stress and then you redo it and then something's different, except then he found that he could apply a stress in the opposite direction. So this would be on the left-hand side of the curve, apply a stress there for 400 seconds, now read out again, and then could see an asymmetric response in the opposite direction. So this means that we can drive reversible changes in the nonlinear stiffening of the material in sort of a reversible way. So this is sort of like inducing magnetization and then inducing magnetization in the other direction, like so flipping the spins. But now in this case, we are doing this with a rheological measurement. Yeah.
How like robust is this anisotropy if you keep cycling?
That's it. Yeah, so we did it a lot, right? So we could go back and forth and ask like how many cycles we could go through. And for this particular system, it was pretty robust. It was surprisingly robust to us that you could actually reverse it. Yeah. Yeah, that's so okay. So I'm gonna, in the interest of trying to get through something I know we've already gone through something, so that's good. But yes, the microscopic structure is changing. So let me, so it's reversible. It's retaining this information about what the most recent strain history was. And so let me just give you sort of the idea of what's happening in the microscopic. So this was a wonderful collaboration we had with Alex Levine at that time and his student, where they built simulations of this. And so the way we can understand the stiffening is through a shear-induced alignment, pneumatic alignment of these polymers, okay? So the idea is that there's some pneumatic order. If you have a certain amount of pneumatic order, then if you now apply a shear strain in that direction of the pneumatic order, that will have a larger number of springs in that direction that can contribute to the spring constant in that direction. Okay, so, but the cool thing is, again, like that pneumatic order, the fact that we can actually have a shear-induced pneumatic alignment is because there's a finite off rate of these crosslinkers microscopically that shear stress allows you to have rearrangements at these smaller scales. If everything was permanently cross-linked, that wouldn't happen. So there's these underlying molecular dynamics that are occurring that are allowing you to have that shear-induced alignment. Yeah. Yeah, there's nothing else here. Yeah, there's nothing else here. Yeah. And I will say that we did this initially for one of these crosslinkers that's very long and flexible and floppy. And we've now tried to do it for other crosslinkers. It's actually very hard to get something with this kind of mechanical memory with other kinds of crosslinkers that are more rigid or have different kinetics or anything. I'm going to go there first. I don't know. Yeah, there. Yeah, I'm happy to talk about that. Yes. Yeah, that's a mode of stress relaxation. It turns out that like I had Patrick McCall when he was in my group was looking at treadmilling. With the crosslink networks, it's really hard to see the stress relaxation in crosslink networks, but when in entangled solutions, you absolutely see it. Yeah. Yeah. Okay. Yeah. 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 I have those slides. A little bit longer. I mean, yeah. So there's a whole dependence of like how long the training history is. It turns out the amount of stress that you need to apply has to push it slightly into the non-linear regime. So a linear stress won't be enough to be able to drive this rearrangement. And the duration does need to be, we have less handle on what controls that time scale. Like I wouldn't say that we have a really good understanding of that. I mean, but it lasts as long as you I mean, it lasts without an applied stress in the other direction. It lasts actually I think it lasts for a very long time. Yeah, sorry. Without erasing the memory, it lasts for a very, very long time. Yeah. Oh, I have a really funny story associated with this project. So like when we submitted the paper, I was an assistant professor, I think at the time, and I tried to call it mechanomemory. And then some reviewer was like, you don't know what you're talking about. This isn't memory. And you shouldn't use words that you don't understand. And and I will say, Ayanna can appreciate this. So Sid Nagel at the time who's a very famous faculty member at Chicago, started using the word memory in his soft matter systems. And he was able to publish lots of papers using, using the word memory. So I don't know, just when people are kind of mean to you, just like I had to change the title of the paper, and and you just have to go with it. And you get treated differently, and it's okay. You just keep moving. Like, yeah. Okay, so there's this stress-induced anisotropy, and Alex and his student were able to basically build a bunch of simulations where they probe the mechanical response by adding in a certain nematic order, that's S here, and calculate with no free degrees of freedom of how the spring constant would change if you induced this certain amount of nematic order. We were able to infer, like from our strain measurements, from this residual strain, how much nematic order was still in line. Like you imagine shearing a sample, and the nematic order sort of is telling you how much shape transformation has been happening in the system. So we were able to take that as a measure of a residual nematic order. And from that, we were able to, I'm going to just go through here. Let's go through here, because this is the exciting graph, where we were able to take our data points. This is all the data points here. This is Alex's and Lewis's theory of how the mechanical anisotropy arose if you have some induced nematic alignment, okay? So microscopically, these cross-linkers are binding and unbinding. The shear is inducing a rearrangement in a nematic order in this gel, and that by changing the shear direction, you can change that. And this is inducing some kind of memory of the past straining history, okay? Okay, okay. All right, so that's this one little crossover point here where you can start to get mechanical adaptation. All right, so for those, yeah. Yeah, I know, yeah. Yeah. I know, you can ask Ayanna about that. No, it's not exactly the same. No, it's not, because that one is about local rearrangements. It's not about the whole mechanical response, but Ayanna and others in his group have been looking at how do you change network elasticity through other kinds of training. It's a little bit different, I mean, than that, but yeah, that was sort of the origin of how we started thinking about these things. I mean, it was exactly, yeah. Yes, in the gel, yeah. Yeah. If you're a good experimentalist, it's not there. No. If you're good at you try to do it so you don't induce any kind of, it should be isotropic beforehand. Yeah, you can't, I mean, you can definitely get anisotropy there, but you try to prepare a sample so it's isotropic.
There regions based on what it is thematic?
Not really. It'd be, I mean, not without some other external field. Yeah. Yeah, no, I know. You can't do that with the gels because they're all kinetic, right? Everything is just driven by the assembly kinetics. You can't, I mean, I guess if you wait, you can in theory wait for years and years and years, and maybe you could get to some kind of, they can't rearrange. It's not experimentally accessible.
You have like side chain that they can align even if the backbone is landed.
Yeah.
Kind of like a second.
Yeah, you could probably get something that's like a nematic elastomer with, I mean, you could probably get something like that. Yeah. That's right. Yeah, well, I've never seen anything like that. Okay, thank you for asking questions. Okay, for those of you who like thinking about gels, yes? You want me to share it? This one? Oh, yeah, Majumdar is the first author. I can give it to you if you want. And Soft Matter, I think in 2015, and I'm the last author, and Levine is also a second to last author. Okay. And then we had another follow-up paper. Actually, we have another follow-up paper recently with Aaron Dinner, and then Stephen Redford was also on that paper. So and that's another Soft Matter paper. Okay, so for those of you who love gels, does anybody here really like gels? Like kinetics of gel formation. I just want you to tell me what you think would happen. So we've been talking about this area. This is where you form space spanning networks. What's supposed to happen over here? What? These are gels. These are kinetically, remember I told you that these are all determined by the kinetics of assembly. What happens in a gel if you don't have enough material to span space? Why would you think it's a liquid? It's a gel. It should be some kinetic cluster, right? It should be some gross non-space spanning. You just gave away the punchline, is why I'm annoyed. Yeah, it should be some cluster of polymers and crosslinkers that is not space spanning, right? They should be some dilute suspension and like it should be small clusters, right? So if anybody knows the history of like colloidal gels, like if you back off the concentration of or the interaction potential of the colloidal gels, you just get small clusters that are diffusing around in space. Okay. When she was a postdoc in my group, I'd never really thought to look over there, right? Who wants to care about small clusters of aggregates? But when she was a postdoc in my group, Kim Weirick did this experiment for some different reason. And this is what she observed when she pushed her concentration space down. So what you can see is this is over about 40 minutes of time. These are actin, that's a fluorescence you're seeing is actin filaments. And you can see these football-shaped objects growing over time, okay? So these are the types of structures that are forming that are diffusing in space. You can see they are small clusters, but they don't look irregular in shape. They have a very, very regular shape. They're also diffusing around in free space. Hopefully this reminds you of those tactoid liquid crystal droplets that I showed you earlier, right? So that's what they are, right? So, and this is again with one particular type of crosslinker. So when you look at each one, there's a characteristic shape of these that you can define. So this is a tactoidal shape that you can circumscribe inside a circle, right? And so if you look at sort of the distribution of this tactoidal structure, they are very peaked, independent of the overall major axis length, right? So the aspect ratio is very well conserved in this, which is telling you a little bit about their what it's telling you about their physical properties of those liquid crystal droplets. Okay, yep. This is a confocal plane. It's a single confocal plane. So there's diffusion in and out of the plane. There is some crowding agent that's keeping everything down to the surface, but there is rotational degrees of freedom here. So there could be some uncertainty with that here. Okay. Okay. All right. So now I want to distinguish them to the tactoids that I introduced earlier, right? So before I told you there was this nice isotropic pneumatic phase transition only when you had short actin filaments, nothing else around at about 500 micromolars of actin, right? What we're looking at here is this 250 micromolars, right, is now 100-fold lower in concentration. This is a dilute phase of actin, and this liquid droplet is being formed because we have a particular type of crosslinker around that's inducing attractive interaction between these actin polymers. Okay, so instead of being a droplet of pneumatic droplet, a liquid crystal pneumatic droplet that's formed or surrounded by a dense isotropic phase, this is a liquid crystal droplet that's in a completely dilute phase. Okay, so it's really a phase separation. All right, it's really driving a droplet formation of this dense polymer liquid crystalline phase. And what I think is super cool is like, you know, I mean, when we think about phase separation, right, when you are working with biological molecules, you have structure of those biological molecules, including filaments, and those filaments are going to impose a structural order to the phases that they create. They're not just spherical spheres, right? And so when you have polymers, now this is inducing a nematic organization within those liquid crystal droplets. Yeah. I can show you that data at the end if I get there. Yeah. Or we can talk about it. Yeah, we did that. It makes the droplets divide. Yeah. They self-segregate in the middle and then they divide. They don't have, I mean, these are too small to really go on the surface. They really go into the bulk and then they just like pinch off and they divide. Yeah. Yeah.
But then in this part, they're forming these very ordered shapes.
Yeah. So I'm here. If I go back here, this is very general, right? This is true with any kind of crosslinker. I'm now talking about this phase, which with certain types of crosslinkers, you're going to get those gross clusters that I just talked about. With some types of crosslinkers or some types of attractive interaction, you can get liquid drops. I should be more precise. So this is going to be this region of the phase space is going to be things that do not span space, small clusters that do not span space. Those clusters can be liquid or solid in nature. Okay, we have one kind of crosslinker that makes something that's like a liquid drop, but it's cool because I never thought that actin in crosslinkers could make a liquid. It was super. I never really considered it as a liquid, like a nematic liquid phase with crosslinkers. That's yeah. Okay, so why don't we hold that question for a second? We don't have direct visualization of that, but we have some evidence that I can talk about. So there was another question, yeah? Yeah, they're like 3D footballs. Oh, that's like the same type of question. I mean, we don't have high resolution information about what the orientation is. I mean, we assume that at the edges there is splay distortion here, right? And that they're going to be dominated. I mean, that's this. I mean, that we do here. Let me show you what when I get that in 2 slides, okay? Okay. Okay. No one actually asked me to prove that they are liquids, but since the liquid group, the people in liquid fields wanted to show liquids, the way you show it's a liquid is you can show mobility of internal components. So you can frap it, you can bleach a region, look at exchange. There is pretty good exchange of the polymers so that there's rearrangements. Okay, so they look like liquid, but even more proof that they're liquid is that they grow in size. Okay, so the average size grows, and the way they grow is by droplet-droplet coalescence. So here is an example, and I wish I had the movie of this. It's like you see these 2 little droplets coming together. They sort of like find each other by the ends, and then they coalesce and then form a bigger droplet with the same aspect ratio. Okay, so these droplets grow by droplet-droplet coalescence, not through accretion of matter from the surrounding bulk. Okay, all right, good. All right, nematics. All right, so before I get to the shape, I'm just going to say so how do these crosslinks actually form a liquid phase? So they form it because the crosslinks are very sparse. There's a low density of crosslinks. If you have a polymer, there's only like about 1 or 2 crosslinks on any polymer at any given time. So there's a lot of flexibility. We also think that the physical properties of the crosslinker are really important to allow for lots of flexibility. Okay, but that's about all we know. We actually don't know what is necessary, what the physical or the physical chemical requirements of the crosslinkers are, or that attractive interaction is to induce a liquid phase, which I think it's a really cool question to ask. There's other cases of other kinds of disordered proteins that can induce liquid phases of biopolymers, but really understanding how you get these composite liquids is something that I think there's a lot of things to explore, both through theory, but also experiments. Yeah. Yeah. Yep. 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 They would be individual polymers just diffusing around. Yeah, there'd just be a gas of filaments. That's correct. Oh, that's funny. That's a liquid in the bulk. Yeah, so it's just, but the physical property would be dominated by the background solvent, right? It's just a gas phase, right? Yep. In a liquid, yeah, but that would be a gas phase of actin. Now this is the actin, the actin-rich phase is forming a liquid, where the actin polymers are relaxing and acting. It's forming a pneumatic. Yeah. Yep, that's right. So, but in the pneumatic itself is a liquid phase. Yes. The tactoid itself is a liquid crystal phase. Yeah, that's what because the crosslinks are transient and allowing enough flexibility. So it's adding enough attractive interactions between the polymers to support some kind of cohesion. But there's enough rearrangements to allow for rearrangements within that phase. So they coalesce, they form an equilibrium shape of a tactoid. It's consistent across all different length scales. Everything is consistent with it forming a liquid crystalline phase in that cross-linked actin. We've only been able to see it with this one type of, well, we've seen it with two different kinds of cross-linking, but yeah, we don't know all the requirements of the cross-linkers to support this liquid phase. It was a surprise to us to see that liquid phase. Yeah. Well, because there's this exchange of polymer that inside that droplet. So this was what I went over right here. So if you frap one side of the droplet, there's exchange of the fluorescence intensity across it. So that means there's mobility over short time scales within the droplet. And then the coalescence is another support of the liquid phase. And then the fact that you see a equilibrated shape across all droplet sizes also shows to you that the shape of those tactoids is dominated by liquid crystal physics, basically. That's what but it was a complete surprise to us and there's other ways we can form this, but it's kind of rare in some ways. Okay. I've also been, I know none of you, I don't think there's any biologists in the audience, but I've always been obsessed with the idea of knowing where these might exist in nature, and I haven't seen any examples of them. So it's been like a fun material science artifact in our group. Okay. So I guess we've sort of touched upon this, but the tactoid is the equilibrated shape for a pneumatic liquid droplet, right? So, and you think about this football shape as some competition between isotropic surface tension, which would tend for you to form a sphere, like a spherical drop, to a cylinder, which is what the preference would be for a pneumatogen, right? You'd want all your rods to align because that gives you the maximized free degrees of freedom of your rod, okay? Now, going back to the shape thing, so as you tune yourself between your isotropic and your anisotropic elasticity, or your your surface tensions, you could imagine tuning the shape of these droplets by making them more pointy and more round. Okay, now again, I'm gonna just keep selling biopolymers as cool materials, is that what's super cool again is that now we have knobs. We can control the length of the polymer distributions, and we can also control the kind of interactions. So like in normal liquid crystals, you don't really have a good control over this stuff, but we can actually do this. And so when we go through and change the amount of cross-linking, we can change the shape of these droplets by making, and it was a little bit surprising to us that with this one crosslinker, as we increased the concentration, it actually made them more round, meaning that this crosslinker is contributing primarily to the isotropic surface tension rather than the anisotropic surface tension. But if you then increase the length of your actin filaments, as you might expect, that's increasing your tendency to want to have more pointy droplets because it's increasing your anisotropic surface tension because you have longer rods. And it also is it's only occurred to these little droplet sizes, which are on the scale of 5 to 10 micrometers. If you make your droplets bigger and bigger, these pneumatic effects becomes less and less important, and your droplets become rounder and rounder. Okay. Yeah. What'd you say? Well, that's a measure of the fact that you're building a liquid out of these anisotropic components, like the rods. Yeah, that's what somebody else asked about anchoring energy. I mean, do we have a good sense of anchoring at the surface? It is something we discussed. I vaguely recall. I would need to think about it more. Yes. Yeah. That's what I think it's this question of anchoring energy, and I don't have a good answer to that right now. Yeah. Yeah. Kinjal definitely thought about that quite a bit. Okay. Okay. Um, okay. So, I didn't get to add activity at all. It's so sad. It's okay. All right. And, it's not about me, it's about you. Um, okay, so it was a surprise to us to see this liquid phase. I think there's lots of really intriguing things to learn about this liquid phase. Since there was a question about motors, I can show you that data pretty quickly. But then also this transition between this liquid phase and this kinetically arrested bundle phase is also something that's super interesting. So as you increase your actin filament length, you get these nice threads. All right, this is just over time. This is a movie that Kim took. But now you can start to see this is just actin and crosslinkers. Over the course of 40 minutes, you start to see that like they start to coalesce and coarsen. You can start to see some tactoid shapes popping up in this. There is a viscoelastic relaxation. All these threads want to become tactoids, want to become round. This is like the relaxation to the equilibrated liquid crystalline tactoid drop of what's happening over this course of this movie. Which again, the fact that you could capture relaxation dynamics of this scale was super cool to us. Then there's a few observations that I just want to make. Yes, Marcus? Yeah. Yeah, so then it makes, yeah, does it, and it collapses like that? Yeah. You do see toroids with actin, not often with just these crosslinkers, but you definitely see that in some cases that then sort of collapse. I mean, okay, so I think this is important because one thing I hope you notice over the course of the movie, if I can go through the movie quickly, I don't know if I can't, it will let me or not. No, it's not gonna, I'd have to play it. But you should see that the length of these bundles is changing, right? So if you just track one of these bundles over time, it starts from being very elongated to more compact, right? So this bundle is shortening over time. And all that's happening is it is driven by surface tension. There's nothing active going on here. It's just trying to equilibrate to its equilibrium shape because it's a liquid. And it's a very slow relaxation time. It couldn't generate a lot of stress, mechanical stress, because it's this very fragile liquid. But it has an interfacial tension that is relaxing towards a droplet. The other thing that's cool is that these polymers, these actin cross-linked has characteristics like other polymer fluids, including Rayleigh plateau instabilities, which happen when you look at the drop breakoff of polymer necks of jets of polymers. And so you can see this purling instability that's happening. And we see that same kind of instability happening in these liquid threads. Okay, so there's a lot of cool morphological dynamics that are just happening in these liquid crystalline phases that are occurring because now you're taking something that I've always thought would be like a kinetically arrested state and allowing it to go transition into something where you are really dominated by surface tension and can equilibrate your shape. All right, that was a surprise to us. Okay, good. Yes. Yeah, no. Oh, so what's happening here? I mean, a lot of times what happens, when you are assembling, like when we assemble this, we're starting the system out of equilibrium, right? So we're polymerized very quickly. And they coalesce into these bundles. That is a unstable configuration. So I think the interfacial tension, like the mechanics dominating, is staying the same, right? The components are not changing over time, but it is just having a slow relaxation to a calibrated shape, which is the tactoid that it wants to be. Yep.
So, like in this problem is, we will be seeing some sort of a gradient to the surface tension, which is like just to understand more clearly from applying any flow fields across the interfaces, or changing this, or like what's the origin of this?
Well, I mean, so the anisotropy comes from the fact that you're building a liquid out of rods. Right? You're building, so that if you have a rod, right, there's going to be a favorable configuration to having all those rods aligned, right? They don't, so that's sort of the microscopic origin of some effects of anisotropy, of desiring to have any elongation, any kind of elongated shape. That's not a round sphere. If you make those rods shorter and shorter, then it becomes minimal. Okay. Okay. Okay, that it concludes all the passive mechanics part, and now it's lunchtime. Since there was one, oh wow, that's a lot of things. Wow, that's a very I would be happy to talk to anybody about enzymatic activity, including molecular motors, since you have been hearing a lot about active stresses. I guess my hill to die on as an experimentalist who has to actually build these things is that when we introduce active stresses into materials, what we're actually doing is introducing biological molecules that spend sort of part of their time acting like crosslinkers, part of their time moving filaments past each other. They are not, in the absence of a surrounding mechanical environment, inducing an active stress. Okay, the stresses are a result of the mechanoenzymatic activity, and what stresses you get, and like what what kind of cross-linking you get, all depends on the actin network, the kinds of motors you have there, and this really affects the kind of emergent dynamics that you see in the system. So we've thought a lot about that, about how do you actually introduce biological, like mechanoenzymes, and how does that affect your material properties to build patterns and forces and fluids and gels that can contract and pattern. And so I'd be happy to talk to any of you about that. Okay. So I'm not going to talk, I'm going to stop there and we can enjoy lunch, and we'll see you at 2 o'clock. Okay. I guess I could ask any final questions. No? Okay. At least I ended at a reasonable stopping point.